So we are, in our unit, we've talked about meiosis. So the process that produces what? Gametes. So we'll move on and talk about next what happens to those gametes. So in sexually reproducing organisms, meiosis produces gametes, specialized sex cells that are haploid. They have half the number of chromosomes as a typical body cell. And so in order to reproduce, fertilization needs to take place. And so fertilization is just the combining of sperm and egg cell. And so in fertilization, two haploid cells combine and form what? Mom? A diploid cell. And we have a name for that diploid cell. Does anyone know the name of the fertilized egg?
Zygote.
Yeah, it forms a zygote. So when male and female gametes unite, they form a zygote. The sperm cells find the egg cell based on sort of chemical signals. And once fertilization has taken place and the zygote has been produced, the next step is for that zygote to begin growing into an embryo and eventually a fetus, or depending on what type of organism we're talking about. And so that process that takes a single fertilized egg cell, a zygote, and turns it into an actual organism requires many mitotic divisions. So it's mitosis, after fertilization, it's mitosis that will form that embryo. So the series of initial cell divisions through mitosis is called cleavage. And it starts from a single zygote going through mitosis and forming two cells. And then those cells reproduce, forming 4, and those all split, forming 8, and so forth. Okay? And eventually we form an embryo. And so the stages in development, the first stages is called the morula stage. This is when it's just a solid ball of cells. Eventually that solid ball of cells forms a hollow ball of cells, kind of like if you've ever seen the inside of a tennis ball. That's sort of what it's like. That's called the blastula. And at this time, all of those cells are identical to each other. They are what we call undifferentiated. They haven't taken on any special role, any special task. They're not specialized in any way. From that blastula stage, the next step is that an indentation begins to form into that hollow ball of cells. And that eventually gets larger and larger. Now, any idea, what does this indentation become? It's going to eventually work its way all throughout this whole embryo. Any ideas? No? It's basically a tunnel through the embryo. Do we have a tunnel through our body? Nope. Yeah, it's the digestive tract, right? The digestive tract. So once this starts to happen, once this gastrula forms with this indentation, this is the point in which cells start to take on specific roles. 3 layers start to form. They're called the ectoderm, mesoderm, and endoderm, outside, middle, and inner layers. And those layers eventually start to become different parts of the organism. If we look at this diagram here, this shows a little bit later stage of development, past the gastrula. This outer layer of cells, the ectoderm, Those cells will eventually become cells of the nervous system, skin cells, epidermal cells. The middle layer becomes skeletal muscle, red blood cells, and a bunch of other things as well. And then the inner layer becomes cells of various organs and so forth. So at this point, the cells are starting to specialize. We call that differentiation. The cells are taking on specific roles. Here's what it actually looks like. So this is a cell in a petri dish probably. This is a zygote, a single cell made by a fertilized egg. So in vitro fertilization, doctor does this in a lab. They can take a sperm cell, insert it into an egg cell, and fertilize it right there. And the egg cell, the zygote then, will start to divide into an embryo in the lab outside of the body. So we have a single cell, then the cleavage begins, start forming 2 cells, 4 cells, 8 cells. Eventually, they're sort of indistinguishable. We have the little marula stage where you just have a solid ball of cells. It starts to become hollow. And here you see the beginnings of that gastrulation, the indentation that's eventually going to work its way through the embryo, forming the digestive tract. So those are the early stages. Now, these cells in these stages are a special type of cell called stem cells. These are embryonic stem cells. Have you guys heard of stem cells before? Some people might. If you have, in what context have you probably heard about stem cells? Dean?
To like try and like regrow some like plastic or something.
Yes, stem cells are a current area of research in which doctors are trying to use these specialized cells to treat a variety of conditions. Stem cells are cells that are undifferentiated that have the potential to form any other type of cell in the body. So a single stem cell can become a cardiac muscle cell, can become a skin cell, a neuron, can become any other type of cell within the body. And so in medicine, these stem cells can be used to treat certain types of degenerative diseases, diseases in which body's tissue is breaking down or damaged in some way. If we can regenerate these damaged cells and tissues using stem cells, then we might be able to treat some of these diseases. In fact, some diseases are already being treated using stem cells. So it used to be, I don't know, 10 years ago maybe, the only way to get stem cells was through creating an embryo and then taking those embryonic stem cells from it. And eventually that was sort of discouraged by federal funding because it involved the creation of embryos, and that was somewhat controversial. Since then, research has come a long way in the last 10 years or so. Now, embryonic stem cells are not necessary for most of this research. Scientists have learned how to take a cell from a human or another animal and basically turn it into a stem cell. Often they do it with fat cells or skin cells, epithelial cells, and they know exactly how to sort of turn the cell into a stem cell. And then that stem cell can be used to create any other type of cell that's needed for certain medical treatments. So this is used, so the process of doing this creates what's called induced pluripotent stem cells. So a pluripotent stem cell is just a stem cell that can turn into any other cell. Induced means they're turning a formerly specialized cell into the stem cell. And when I went to that, the Masonic research facility I told you about a couple weeks ago where they were doing a lot of cardiac research. So they presented like 10 different researchers presented to us and and told us what they're working on. Every single person that presented, their research required induced pluripotent stem cells. They all started with that. What many of them would do is take a patient that had, let's say, a rare genetic cardiac disease. Okay? Obviously, they're not going to experiment on that person. But they can take a sample of their cells, turn it into a stem cell, then take that stem cell, turn that into cardiac cells in a petri dish, and they have a tissue sample that has that genetic disorder, and then they can use that to develop treatments, okay? To try to look at the genes and see what's causing it, to screen medications. So they can do many things once they have those cells. So all of those research projects needed iPS cells. Also, stem cells are currently used in treating Parkinson's in some cases, in treating macular degeneration, which can lead to blindness. Now, there are many places that claim to be using stem cells for other things that are not proven. And so that's something that has to be further researched, some of these other forms of treatment. So here's the process. Take a donor or a patient, take a sample of their cells from, let's say, their skin, reprogram them. Basically, they use different chemicals, different environmental factors to change those into iPS cells. These iPS cells can then be turned into any other type of cell in the body. Scientists have learned, okay, if we give these stem cells these chemicals in the petri dish, they will turn into neurons. If we give them this other set of factors, they'll turn into cardiac muscle cells. And they've learned how to do this. So basically they can turn these cells into anything else. They can then, like I said, look at correcting the genes, look at screening for different types of medication, trying to fix the damage and then retransplant them into a patient. These are all possibilities using these iPS cells. All right, let's just watch this video quick. Any questions about stem cells? All right, so let's talk a little bit about animal reproduction. We'll start by talking about external embryonic development. So in most types of animals, the reproduction happens in the water. Happens using what we call external development. So most aquatic organisms, animals that live in the water, release eggs into the water that are unfertilized, and then typically males then release sperm into the water, and then those sperm cells can make their way to the eggs and fertilize them. Usually the cells, the egg cells, they don't have any sort of covering on them. Sometimes they're in like this jelly-like coating to keep them together because they need to allow the sperm cells to reach those eggs to fertilize them. Usually organisms that have this process of external fertilization produce lots and lots of eggs. Fish can release hundreds of thousands of eggs. Amphibians, hundreds of eggs. Because many of them don't survive. Many of them are preyed upon. Many of them don't get fertilized or just never grow. So you need to release a lot of eggs just to provide for some that actually hatch and grow into adults. Most animals that reproduce like this, they have behaviors to synchronize. It's not like when they reproduce, fish just randomly release eggs into the water and males just randomly release sperm into the water. Usually there are behavioral cues that the male and female are in the same area and release their reproductive products at the same time. Like we call it spawning in fish. Inside of that egg is a yolk as well that provides the energy for that embryo to grow. And typically water, oxygen, waste can just diffuse in and out because they're just sitting there in the water. On land, other adaptations are necessary. Because these same types of eggs, if they were laid on land, they would just dry up. So land animals had to develop other means of reproduction to allow them to reproduce on land. And so one of those adaptations is that fertilization in these animals doesn't happen outside of the body. It happens internally. So males deposit sperm cells into the female reproductive tract. Those sperm cells make their way to the egg to fertilize it. Because they don't have that watery environment to have external fertilization. Once fertilization has taken place, these organisms then lay an egg. And really what an egg is a tiny little encapsulation of a watery environment that can provide the growing embryo with everything that it needs. So those eggs are waterproof so that the egg doesn't dry up. Inside of that egg is also the yolk to provide the energy that the embryo needs to grow. There's liquid, there's protein. There's basically all of the things that the embryo needs to grow are contained within that egg. There's respiratory structures to take in oxygen, to release waste. It's all inside of the egg. And so this happens in insects release eggs, reptiles, birds produce eggs, obviously. And this is one means of reproducing on land, producing these waterproof eggs. And some mammals still have this feature. Primitive mammals, the monotremes we call them, they lay eggs. Okay? Things like the platypus and the echidna, they release eggs, but that's only 2 species. Most mammals have internal development. So rather than laying an egg after fertilization, mammals have evolved to keep the young internal, and the embryos grow into the young inside of the female. Now marsupials, they have internal development. However, they don't have a placenta to provide the growing embryo with energy and oxygen and so forth. Instead, they give birth to very immature young. So the embryo uses a yolk to provide the energy to grow from a zygote into a tiny little organism. It's born extremely premature, and then it makes its way into a pouch and stays there to sort of finish its development until it can become independent. Inside of that pouch are the mammary glands where the young marsupial can nurse, getting energy until it grows large enough to be independent. So examples of marsupials, koalas, kangaroos. In North America, a possum is a marsupial. What you're probably most familiar with are what we call the placental mammals. Placental mammals includes humans. And so they have, again, internal fertilization. Sperm meets the egg cell inside of the female. And then the young develops internally as well. And it stays in the uterus, or we might call the womb, where a placenta forms. So placental mammals form this organ called the placenta. That's extremely important. So during the initial stages, after the zygote's produced, to provide the energy to divide and grow, there is a very small amount of yolk to provide that energy. Once that embryo makes its way into the uterus, it attaches to the wall of the uterus. And that's where the placenta forms. So the placenta is an organ that's basically filled with blood vessels. It forms from a combination of maternal tissue from the mother and embryonic tissue from the embryo. And in the placenta, capillaries from the mother are right next to capillaries from the embryo. But blood does not go from the mother into the fetus. That's a common misconception. The blood supply of mother and embryo and fetus are kept completely separate. There's no sharing of blood. But because these capillaries are right next to each other, that allows for diffusion of materials to take place. So as the mother takes in oxygen, goes into her blood goes to the placenta, some of that oxygen can diffuse into the blood of the embryo, providing with oxygen. Same thing with nutrients. As the mother eats, nutrients from what she ate travel through her blood to the placenta, where those nutrients can diffuse into the blood of the embryo. Material goes the opposite way as well. Waste products that are generated by the embryo can diffuse out into the mother's blood, and she can then excrete them in the lungs and the kidneys and so forth. So the developing fetus never eats, never drinks, never breathes, never urinates, never defecates while it's in the womb. Doesn't need to do any of those things because basically the mother is doing all of that for the fetus. The first breath will be after the fetus is born and the placenta detaches. The umbilical cord connects the fetus, embryo, to the placenta. It contains 3 blood vessels, 2 arteries that carry the fetal blood out to the placenta, and 1 vein that carries the blood back. So we'll look at some pictures here. So these are the monotremes, the egg-laying mammals. This is the echidna, sometimes called the spiny anteater. This is it before the spines form. This is the platypus, the duck-billed platypus, another monotreme. These are the only 2 monotremes. Okay? They lay tiny little eggs which hatch in the immature young, and then they nurse to finish their development. These are marsupials. Kangaroo, koala. These are opossums, wombats, and there's others. So they, like we said, give birth to very immature young, which finish the development in the pouch as they nurse. I think you guys saw this in 7th grade. This is marsupial birth. Should we watch it again or should we skip it?
Yes, watch it.
All right.
You see like an elephant? There's someone.
All right, so if we just placental mammals include probably the vast majority of mammals that you are familiar with. So this is what a placenta actually looks like. So attached is the umbilical cord, and it's basically just a collection of blood vessels. This was attached to the uterus in the mother's womb. Here's when it's attached. You can see the blood vessels. Basically, they bring the blood both ways, but that blood enters through where your belly button is now. That's where, once your umbilical cord connected, it's filled with blood vessels that lead into the circulatory system of the developing embryo and fetus, so that the materials that are diffusing in are delivered to the fetus, and the waste products are delivered out and can diffuse into the mother's blood. This is a sort of close-up just after birth of the umbilical cord. After birth, it gets cut, tied off, and eventually heals into the belly button. All right. Nope, not what I wanted. All right, so just to finish up, I guess we'll finish up with this slide. The male reproductive system, okay, is specialized for producing sperm cells and then delivering them into the female reproductive tract. So now we're referring to humans. Humans have, males have 2 testes that produce sperm cells. They also produce testosterone. The testes are where the male sex hormones produced. And testosterone is also, besides developing male sex characteristics, testosterone is responsible for the production of sperm. So the sperm cell's meiosis takes place in the testes, in a part called the seminiferous tubules. That's where diploid cells make haploid sperm cells. Those sperm cells then move into another part of the testes called the epididymis. This is where they actually mature from their initial form. develop the flagellum and the parts they need. So in humans, sperm are produced most efficiently at a slightly cooler body temperature than body temperature. So they're held outside of the body in the scrotum, which keeps them, a couple degrees cooler than internal body temperature, so that sperm cells can be produced more efficiently. And in development, in utero, the testes are in the abdomen, in the abdominal cavity. Around the time of birth, they descend into the scrotum afterwards. So the sperm cells that are produced in the testes, in the seminiferous tubules, are really, really small, especially compared to the egg cell. An egg cell produced in females is one of the largest cells in the body. Sperm cells are very small. Basically, just are large enough to contain the 23 chromosomes that they need to carry. They're produced, like we said, and then mature in the epididymis. When they are released from the male's body, they travel through a tube that's called the vas deferens, which merges with the urethra, okay, and then out of the body. So at one time, a male could release 40 to 300 million sperm cells at one time. And they can survive for 24 to 48 hours. And the sperm cells are surrounded by a fluid called semen that is made up of several different things. Okay? There are several glands that produce the components of semen. The prostate gland, for example, that I'm sure you've heard about. This produces a fluid that helps to neutralize the acidic vaginal fluid so that the sperm can survive. They couldn't survive on their own in those acidic conditions. So the prostate gland produces neutralizing agents. Cowper's gland produces fluid for moisture. The seminal vesicle produces fructose to nourish the sperm. They have a long journey for a very tiny sperm to make. They have a long journey, okay, to reach the egg. So they need some source of nutrients, and that comes from this fluid from the seminal vesicle. And during ejaculation, they're released through the urethra from the penis, and then begin their journey to make their way to fertilize the egg cell, which eventually will happen in the ovum. So if we look, this is sort of close-up of the testes, the microscopic structure. So these little wound-up tubules, that's where meiosis is happening. They make their way then into this outer layer. This is the epididymis, where they mature, and then they are released through the vas deferens. If we look at sort of a larger overview, you see the testes or testicle means the same thing. The epididymis are on the outside. This is the vas deferens, which eventually, as it travels, we have the seminal vesicle and the prostate gland. The Cowper's glands in there as well that secrete those fluids, and then eventually merges with the urethra and out through the penis. So, you've heard of a vasectomy before? Surgery that a man can have if they don't want to be able to make a woman pregnant anymore. It's basically just the doctor cuts the vas deferens so that the sperm cannot be produced anymore. However, the other parts of the semen are still produced. So we'll talk about female reproduction on Monday.